Hi, everyone. Nice to meet you. This is Zhang, the technical support and the project manager in GemPharmatech. Dr. Guo has some emergencies to deal with, so today I will be the speaker to introduce our immuno-oncology platform, especially some study progresses on NK-related humanized mouse models. So before sharing, I'd like to introduce myself. I graduated from Southeast University and joined in GemPharmatech since 2020. So it's been my first year working as a technical support and a project manager in GemPharmatech. So as you might know that GemPharmatech is a global biotech company dedicated to providing a one-stop solution for all biological communities. We now have more than 1,500 employees, and our core R&D team has over 20 years experience in mouse model generation field, with 6,000 new genetically engineered mouse models being generated every year. We now have a mouse model of over 20,000 for distribution, which makes us the biggest mouse model resources in the world. For the immuno-oncology platform, for the immuno-oncology platform, we offer different mouse models and services. For example, the humanized immune checkpoints mouse models generated based on the immunocompetent background mouse. For example, the humanized PD-1 and the PD-L1 mouse models. The immunodeficient mouse models, such as the NCG and the next generation NCG. The human immune system reconstituted mouse models, such as the human PBMC and the HSC reconstituted on NCG mice, and the next generation NCGs, and also the spontaneous tumor models. So using all these kinds of mouse models, we offer different services according to clients' needs. For example, the bispecific antibody efficacy evaluation, cell or gene efficacy evaluation, and so on. So today in my webinar, I mainly focus on NK cell related mouse models and services. We know that NK cells is becoming a rising star in cancer immunotherapy. Different strategies have been used to introduce NK cell activation to improve the tumor cell killing effect. For example, NK cell mediated antibody-dependent cytotoxicity, the ADCC effect, specific or triple specific killer engagers capable of binding NKG2D expressed on NK cells and the tumor antigen targeting of tumor cells. NK immunotherapies, cytokines such as the IL-15 can also be employed to generate cytokine-induced NK cells. So according to research and the markets, we can see that the NKC therapeutic marketing is fast growing. The compound annual growth rate is more than 17%. So among all these strategies, we can see the ADCC effect and the CAR-NK cells have drawn a lot of attentions. At the AACR meeting, AFMED announced the latest result of phase 1 and 2 trial of its NK cells and AFM30 antibody targeting CD16A and CD13 in CD13 positive Hodgkin lymphoma and non-Hodgkin lymphoma patients. And the ORR, the objective response rate, is 100%, and the complete response is almost 17%. So we could see very promising clinical trial results have been achieved by NK cell-based therapy. But we also know that there still remain much changes on preclinical NK cell therapy studies. because we know that the cytokine IL-15 is critical for development and persistence of NK cells. However, the mouse IL-15 poorly supports human NK cells due to its low affinity to human IL-15 receptor. So different strategies have been used to overcome these limitations. For example, the engineering NK cells, CAR-NK induced with IL-15, or the employment of memory NK cells, as well as the injection of the exogenous IL-2 or IL-50 to support the NK cells in the mouse body. But all of these strategies require the repetitive NK cells infusion and the cytokine injection. But this also enhances the risks of adverse reactions, because we know that the delivered doses of the exogenous cytokines exceed several times the physiological levels. So this enhances the risk of adverse reactions. So we really need mouse models that could have NCG-IL2 or NCG-IL15 expressed at a physiological level to support the human NK cells reconstitution in mouse body. So we could have a good model to study the NK cells function in efficacy study. So in our facility, we offer different strategies to overcome different strategies for the human NK cells reconstitution for our clients. So I'm very proud to show you our immunodeficient mouse model, the NCG mouse model. The NCG mouse model was generated in GemPharmatech. It lacks T cells, B cells, and NK cells, and also deficient in macrophage and dendritic cells. So it's the best model for engraftment of human cells. And here is show the strategy of how we generate NCG mouse model. We use the NP background mice. We know that the NP background mice is deficient in the macrophage and the DC cells. So in this background mice, we use CRISPR-Cas9 technology to knock out the PRKDC gene. So the mouse model will lack of T cells and B cells. And we also knock out the IL-2 gene. So the mouse will lack of NK cells. The NCG mouse model is exclusively distributed by Charles River in USA and the European market now, but it's developed by GemPharmatech. NCG mouse still have some limitations after PBMC or HSC human stem HSC immune cells reconstitution. To overcome these limitations, we developed a lot of generations of NCG. Here is an example. For example, if clients want to have high levels of T cell reconstituted, here is our recommendation. If clients want to have the high level of human NK cells reconstituted in the model, we would recommend the NCG-IL15 or NCG-IL2. So in these two models, we use CRISPR-Cas9 technology to insert the human IL-15 into the NCG background mice. So this mice could have human IL-15 expressed, such support the NK cells development and maintenance. Also, in the NCG-IL2 model, we insert human IL-2 gene into NCG mice. So this mice also support the reconstitution and the maintenance of human NK cells after HSC engraftment. So in the next slide, I'd like to give you some case studies and show you some data we have achieved in our facility or from our users. So here is the strategy of the NCG-IL15. We use CRISPR-Cas9 to use human IL-15 to the NCG mice. After the generation, we do the verification at both mRNA level and the protein level to show that the human IL-15 could be expressed in the mouse body. And we did detect a high level of NCG-IL, human NCG-IL expressed in the mouse body, both at mRNA and the protein level. And after the verification, we also do a lot of efficacy study using these 3 different strategies. We could either purify the NK cells from the periphery beyond the mononucleotides and then inject the NK cells into NCG-IL15. So this model could have a high level of NK cells reconstituted. And also we can do the PBMC injection directly into the NCG-IL15 mice. So this mice could have both T cells and NK cells reconstituted. And what's more, we could also have the CD34 positive huHSC stem cell injected, engrafted into the NCG-IL15. So in this model, the mouse could have both T cells and NK cells reconstituted. This is a comparison table among these 3 different mouse models. So if you want a high level of NK cells reconstituted only, the first model could be our recommendation. So if you want to have both T cells and NK cells reconstituted, then PBMC or huHSC engraftment in the NCG-IL15 would be our recommendation. But just note that in the PBMC engraftment, as we know that the most important problem of this model is the GVHD may happen at an early age. So the window period of the efficacy study is not as long as the other 2 mouse models. So next, I give you some case study to show you some data we collected from one of our users. They published this data in 2021. So what they have done is that they purify NK cells from PBMC first and then engrafted the NK cells into both NCG and NCG-IL15 and do some comparison. First, we can see the reconstitution level. In the NCG model, after the NK cells engraftment, we can see the NK cells level is could only be maintained in the first and the second week, but after the 3 weeks of engraftment, the NK cells was depleted. There is no NK cells anymore because this model can't support the NK cells maintenance. But in the NCG-IL15 model, we could see a relatively higher level of human NK cells reconstituted, and also the NK cells could maintain at a relatively very long time period. So this gives enough the window period for efficacy study that man need human NK cells. And also they do the mice body weight and the survival detection. They found that there's no differences in the NCG-IL15 after NK cells engraftment in the body weight or the survival rate. And more importantly, they want to see if the reconstituted NK cells in the mouse body is functional. So they do some analysis. They analyze some functional markers in the human NK cells, such as the NKG2A, NKG2D. And they found that the NK cells could express these functional markers, and more importantly, they also detected the granzyme and preference in the NCG-IL15 after the NK cells engraftment, and compare this with the purified NK cells and the NCG models. We could see that the expression level of these two proteins is comparable in the purified NK cells and the NCG-IL15 mouse models, but the expression level is pretty low in the NCG mouse models. So this shows us that NCG-IL15 could support the reconstitution of NK cells, human NK cells after the NK cells engraftment, and also the NK cells are functional NK cells. So after that, they also do some efficacy study. What they have done is that they injected their CAR NK cells into the NCG-IL15 models. And we can see that in the NCG-IL15 models, this model could support the development and maintenance of CAR NK. And more importantly is that in the NCG-IL15 mouse models, we could see a significant decrease in the tumor radiance after the CAR NK administration. So this shows us that the NCG-IL15 is a very good model for the NK cell-mediated efficacy study after NK cells engraftment. We also, in our facility, we also have done a lot of verifications and efficacy studies. So here I'd like to show you some data we have achieved. What we have done is to do the huHSC, the stem cell engraftment in both NCG and NCG-IL15. And after engraftment, we could see a very apparent differences between NCG and NCG-IL15 in the human NK cells reconstitution. If we detect the CD56, the marker of NK cells, we could see that in NCG-IL15, there is a high level of NK cells reconstituted in IL15 model, but not in the NCG model. And we also do some detection of the functional markers of NK cells. And more importantly, we do the ADCC effect detection in both in vitro and in vivo. First, let's see the in vitro data. We could see that if we if we capture the NK cells with the tumor cells in vitro, we could see that with the increase of NK cells, the cytotoxicity was increased as well. So that's reasonable. Importantly, if we administrate the rituximab, know that the rituximab have the tumor killing effect, mainly via the ADCC effect. So if we administrate the rituximab, we could see that with the increase of the dosage of rituximab, the cytotoxicity increased as well. So this shows us that in vitro, the NK cells could play a tumor killing effect via ADCC effect. Also, we do in vivo efficacy study in these two different models. First is we do the huHSC engraftment in the NCG-IL15 model. And after the administration of the rituximab, we could see that there is a very apparent obvious tumor killing effect. But if we administrate the rituximab in the NCG-IL15 without huHSC reconstitution, there is no tumor killing effect. So this shows us that the NK cells plays a role in the ADCC effect. So the NK cells is necessary in the rituximab's tumor killing effect via ADCC. So that's interesting. More importantly, another different efficacy study I'd like to share with you is that the NKG2A antibody efficacy study. We know that the NKG2A transfer or inhibitory pathway in the NK cells activation. So if we block this pathway, we could see that the tumor killing effect, we could see a very obvious tumor killing effect. This indicates that the NK cells was activated and plays a role in tumor killing effect. So that's all the data we'd like to share with you in the NCG-IL15 models. So as I have mentioned that we also have another model to support the human NK cells reconstitution, the NCG-IL2 mouse models. So in this model, we inserted the human IL2 into NCG. And here is a strategy show you how we do the huHSC engraftment in these two kind of models. First, we will do the invitation to the mice. And after the invitation, we will engraft the CD34 positive huHSC into the mice. So the mice will have the human immune cells reconstituted in about 8 to 10 weeks. So after that, we'll inoculate CDX or PDX to the mouse models. So the models will have the tumor development. And then we can administrate some positive drugs or test articles from clients for the efficacy study. As well, after we generate the NCG-IL2, we also do some verification to make sure that the human IL2 could be expressed in the mouse model. So we collect different tissues after different tissues in the NCG-IL2 and could detect a high level of NCG-I, could detect a high level of human IL2 in these different tissues. And more importantly, after the huHSC stem cell engraftment in the NCG-IL2, we could have a very high level of NK cells reconstituted. So the ratio is relatively very high, but we need to remind that there is no T cells reconstituted in this model. So very different from the NCG-IL15. As we have mentioned, the NCG-IL15 support both T cells and NK cells reconstitution, but in the NCG-IL2, it mainly supported the NK cells reconstitution. So after the verification, we also do some efficacy studies. Here is the data we'd like to show you. In the huHSC NCG-IL2 mouse models, we inoculate breast cancer cells and do the efficacy study. As we can see that with the dosage increase, there is a dramatic tumor killing effect in group 3, and then no differences in the body weight among these 3 groups. So this shows us that the NCG-IL2 mouse model is a very good model for efficacy evaluation using the human NK cells after the huHSC engraftment. Besides these 2 models, I'd like also to show you that our cell line library, because we know that in the immuno-oncology study, cell lines were necessary or important resources. So in our facility, we have different kinds of cell lines, such as human cell line, the CDX cell line, or the Zhang cell line, or the humanized mouse cell lines. The humanized mouse cell lines means that the cell line is still Zhang cell line, but we have 1 or 2 targets humanized, just to replace the mouse gene with human gene to explore the human targets to some test article. And also we have the luciferase tagged cell lines. Because we have our own cell line engineering platforms, we could do the cell line engineering according to clients' needs. So if you are interested in any of the cell line model, feel free to reach out. We are more than glad to share more data with you. Besides the cell lines, we also have PDX resources covering different tumor indications, such as breast cancer, the pancreatic cancer. For the pancreatic cancer, there were about 18 cancer samples with a wide variety of mutation targets. So for all these targets, we have done verified by vast sequence. So we found that almost covering all the types of highly mutant tumor targets. So if you are interested in any of the tumor indication or the mutation targets, feel free to reach out. We know that for CAR-NK or CAR-T efficacy study, sometimes topic injection is very important. So it's required the live imaging to monitor the tumor development. The in-vivo facility will have generated different kinds of different luciferase-tagged tumor cells. So this make the orthotopic models available. We could do various kinds of orthotopic injection according to clients' needs, such as the brain orthotopic, the pancreatic orthotopic, liver, breast, and so on. Last, I'd like to show you the 6 subsidiaries in China, such as the Shanghai site, Guangzhou, Changzhou, Chengdu, Beijing, as well as our headquarters. So if you have a chance to come to China, welcome to our company. Thank you. That's all for my sharing. So if you have any questions, feel free to type in the chat box. I received the first question. The first question is that we know that the reconstitution of HSC in mouse body require a long time. So if I want to do efficacy study in the facility, do I need to wait for a long time? Thank you for your question. That's a very good question. So I'd like to share with you that in our facility, we will do the reconstitution of HSNCG as well as HSNCG-IL15 weekly. So every month, we have hundreds of HSNCG or HSC-IL15 mice reconstituted well. So if a client wants to do efficacy study in our facility, there is no need for them to wait for a long time because we have the well reconstituted mouse ready for their efficacy study. We can study the efficacy study in a pretty short time. Thank you for your question. So the second question is that what's the difference between NCG and NSG? So thank you for the question. So for NSG, as we know that this is from Jackson Lab. They generated this strain via ES cell targeting technology to mute the IL2R gene and then cross with the LD skid mice to generate the NSG. That's their strategy. But in our facility in GemPharmatech, we knock out the PRKDC, IL2RG on the pure NOD background mice to generate the NCG mice. So the biggest difference, I think, is the strategy. We use CRISPR-Cas9 on the pure NOD background. I think this is also a considerable advantage over strains that had to be generated by crossbreeding mutations that were generated on multiple genetic backgrounds. We use the pure NOD background. But these two strains, both NSG and NCG, are all severe immunodeficient mouse like T cells, B cells, and NK cells. Thank you for your question. So if you have more questions, feel free to type in the chat box. Here, I'd like to leave 7 minutes for you to type in your questions, and we will collect all your questions one by one and get back to you one by one via email after this webinar. Okay, thank you for your questions, and thank you for your time in this webinar. So we have recorded this webinar, and we'll upload this webinar to our YouTube channel, GemPharmatech YouTube channel. And also in the GemPharmatech YouTube channel, you could find all the previous webinar and this webinar as well. So if you are interested in our mouse model services and our company, feel free to go to our YouTube channel. Thank you for your time. So see you in next webinar. Goodbye, everyone.